So we're in the last session in the home stretch here, and so we have 3 exciting talks before the break, and then we'll come back for another 3 presentations. Our first talk is by Siobhan Brady from the University of California, Davis. She's within the plant biology group as well as the UC Davis Genome Center, and she's going to talk about transcriptional regulation of root development using Arabidopsis as a model system. So thanks for coming.
So I'd really like to thank everybody for inviting me here. And so I'm going to switch a little bit in terms of topics. I'm going to be speaking to you about plant roots. But first, I want to remind everybody about how beautiful plant development really is. So on this slide, I have pictures of 2 different organs and 1 cell type in a plant. On the left-hand side, you have a picture of Romanesco broccoli, which some of you may have eaten before. And so basically, Romanesco broccoli is really, really beautiful, and it has a set of these different meristems that are organized in this really beautiful Fibonacci series, or the same kind of pattern that you would see in fractals. The pattern that we're most interested in my lab is that of root development. Tell me if you can see the pointer here. So root development. Roots are really fantastic organs because they basically can dynamically explore the soil, and they can try to very quickly accumulate as many nutrients as they can. And then the cell type that we're really interested in the lab is Xylem cells. And so many of you here in this room are very interested in how this secondary cell wall is composed, as well as how you can extract sugars from the Xylem cell. So in our lab, we're interested in 2 questions. So first of all, what are the regulatory processes that determine cell identity and patterning? And then how are these regulated temporally? Okay, so we use the root as a model developmental organ. And up here I have an image of an Arabidopsis root tip, a cartoon of it. And you can see, or I'm going to show you how the root is actually able to simplify the study of space and time and development. So these different cell types are shown here in different colors. And as you can see, for most of the outer cell types of the root, they're arranged in a rotationally symmetrical fashion. So if you were to take a cross-section, at any point within this root, you would basically end up with a mirror image on the other side. And so this simplifies our study of space, or of different cell types, to a single axis in the root's radial axis. And then at the tip of the root, we have the stem cell niche. And so in this stem cell niche, we have a quiescent center here in gray. This quiescent center is surrounded by these stem cells that are shown in different colors. Each of these stem cells will divide and give rise to a daughter cell. That daughter cell will go on and differentiate into its respective cell types. And since plant cells don't move, these cells are basically constrained within cell files. So with each new division of one of these initial cells, it successively displaces a slightly older cell along the root's longitudinal axis. So what this means is that as the root grows down, you can basically look at a progressive gradient of developmental age. So you can go from the youngest cells in the root progressively up to the oldest cells in the root. So we can look at both space and time in a single organ. So today I'm going to be telling you about how we've been able to use these features to look at the Xylem cell differentiation network in the Arabidopsis root. Okay, so there's a couple of data sets that are available to us that really simplify our studies. So the first is a data set that describes expression in 14 out of 15 of the non-overlapping cell types in the Arabidopsis root. So this data set consists of roots which were protoplasted and then fluorescently active. They were sorted using a fluorescent activated cell sorter. And then we can basically isolate individual cell types or populations of cell types. And then we isolate RNA from these and do microarray analyses. And so in addition to space, we also wanted to capture time. And so we have another data set which profiles expression in developmental time. So what we did was basically took a series of sections along the Arabidopsis root. And again, you move from the youngest part of the root up to the oldest part of the root. The idea here is that with each successive section, you have a different point in a developmental time course, moving from the youngest to the oldest parts of the root. So we again isolated RNA from each of these and did microarray expression profiling. And of course, we then did some computational analyses of this data. And our goal here was to identify co-expressed gene groups in space or in time. And so in this heat map, each row represents a group of co-expressed genes, with yellow showing relative upregulation and blue showing relative downregulation. So there are approximately 51 groups of co-expressed genes in space and 40 groups of co-expressed genes in time. Using other computational tools, we've been able to integrate both of these data sets together. So now you can go online, and for any gene of interest, you can go and figure out exactly where that gene is expressed in the root, in what cell type, and at what time point is it expressed. Okay, so let's go back to transcription factors and transcriptional regulatory mechanisms. So at its very base, what are transcription factors? Just to remind everybody, transcription factors are proteins that bind to regulatory sequences within promoters, and they then interact with the core RNA polymerase machinery to either activate or oppress gene expression. But we in fact know that this is a really overly simplistic view of transcriptional regulation. These transcription factors in fact can act in a combinatorial manner, and they act within these immense, complex, interconnected regulatory networks. They act within these networks in individual cell types, even in individual cells, across tissues, across organs, and these networks dynamically change over time and in response to the environment. And so the work that I'm going to be telling you about today looks at gene regulatory networks, or transcriptional regulatory networks, and we use systems biology approaches to answer these three questions. So which transcription factors regulate gene expression and development? How are these transcription factors interconnected? And which transcription factors are the most influential in this process? Okay, so what kind of data do you need to have to be able to map these transcriptional regulatory networks? You need three pieces of data. So in a multicellular organism, it's extremely important to determine where transcription factors are located. So at what cell types and at what time points can they be found? And so in our lab, we use these microarray data sets that I just spoke to you about, which profile expression in different cell types and different time points. And in our lab, the tissue that we focus on is the Stele tissue, or vascular tissue of the root, which contains xylem cells, which I'll be focusing on today, phloem cells, procambial cells, which give rise to secondary growth, and pericycle cells. So next, you need to determine what targets or what regulatory sequences these transcription factors interact with. And we use a yeast-1 hybrid approach to do this. So we have a collection of transcription factors that are fused to the GAL4 activation domain. We then take promoters of genes, which we've identified to be involved in some biological process. We fuse these upstream of a His3 reporter and to a LAC-Z reporter. If our transcription factor interacts with our promoter, and then we will get activation of His3 and the LAC-Z reporter. And we know that at least in yeast, that transcription factor can interact with the promoter. And of course, that's all in yeast. And so in previous work published this year, we showed that we've been able to validate these interactions that we identified in yeast in planta. So 4 out of 5 interactions that we identified in yeast, we were able to validate as determining physically by chromatin immunoprecipitation. Okay, so next, we need to determine whether or not that transcription factor actually regulates the expression of its target gene. There are many examples of cases in CHIP studies, for instance, where many transcription factors reside on regulatory sequences, but they don't necessarily regulate the expression of that gene. So we use a combination of genetics and conditional induction assays to test these regulatory interactions. And, oh, this slide didn't show up, but in fact, we have 59% of regulatory interactions that we've been able to validate in planta using this method. Depending on the network that you're using at, that number can even go up to 70%. So it's a really robust way to be able to screen for regulatory interactions in planta. So this is what our method looks like. And so we have now a set of 92% of all transcription factors that are expressed in root vascular tissue. These are arrayed out into a matrix fashion. And these transcription factors are further arrayed out in quadruplicate so that you can get 4 technical replicates of an interaction. This is an example of interactions activating the LAC-Z reporter and the His3 reporter. And this is all done in a semi-automated fashion. There we go. There's my 59% of interactions. Okay, so using this method, we wanted to be able to map a regulatory developmental program in both space and time. And so the regulatory program that we're interested in is xylem cell development. And so xylem cell development in the root, xylem cells have to undergo 3 different developmental programs. So first, these cells have to be specified as either a protoxylem cell or as a metaxylem cell. So protoxylem cells and metaxylem cells can be differentiated based upon the secondary cell wall deposition pattern along the length of that cell wall. So protoxylem cells are characterized by this helical or annular pattern that lines the wall, whereas metaxylem cells basically consist of a sheet of secondary cell wall with small little pits in it. So the cells have to be specified first. They then have to undergo the secondary cell wall biosynthesis program, And then finally, these cells undergo their terminal differentiation program, which is death. So in this talk today, I'm just going to focus on these first two developmental programs. And the first thing I want to really emphasize is that this process has to be really tightly regulated in time. So cells undergo elongation prior to this differentiation process, so prior to the deposition of the secondary cell wall. If this program right here, if secondary cell wall synthesis, occurs earlier or precociously in development prior to the cell elongating, then the root will arrest its growth because it can no longer elongate because it has this really thick cell wall surrounding it. So you need to really make sure that you turn this program on at the right time. This process is also really tightly transcriptionally co-regulated, and this has been used quite often in bioinformatics screens. This is a set of secondary cell wall biosynthesis enzymes and their expression in roots in time, going from the youngest to the oldest parts of the root. And you can see that there's 2 peaks of expression in developmental time in the Arabidopsis root. So this tight co-regulation implies that there are common transcriptional upstream regulators in this process. So despite the fact that there must be transcriptional regulators that are common in regulating this process, it's been really, really difficult to identify transcription factors or master regulators that are both necessary and sufficient for the regulation of this process. So single mutations in many transcription factors that have been shown to be sufficient for this process do not show a phenotype in a single loss of function background. So there are 2 different classes of transcription factors which have been shown to be sufficient to regulate xylem cell specification and secondary cell wall synthesis, but not necessary. The first are these NAC domain transcription factors, VND6 and VND7, which, like I said, are sufficient to regulate this program. And the second are these HDZip3 transcription factors, including Revoluta, Fabulosa, and Favoluta, that again are sufficient but not necessary to drive this process. So some really important questions remain in this developmental program, both in the root and the shoot. So first, what regulates the timing of the expression of these regulators that determine xylem cell specification? Next, what are the targets of these transcription factors in terms of enzymes regulating secondary cell wall biosynthesis? And finally, what are the mechanisms that control the timing of this process? And finally, how are the HDZip3 and the NAC domain transcription factors regulated in terms of a pathway? Which one is in charge of which? Okay, so in order to answer this question, we decided to use this yeast-1 hybrid approach. And so my graduate student, Mallory, went through the literature and identified 50 genes which are expressed in the root xylem and which are either involved in xylem cell specification, cellulose biosynthesis, lignin biosynthesis, hemicellulose biosynthesis, or which are co-expressed with secondary cell wall regulators. So she cloned the promoters of all of these, put them into yeast, and screened them against our collection of about 653 transcription factors. So this is the network that she came back with, and so it's a little bit crazy. There's a lot of transcription factors that are binding to these promoters of these different genes. The promoters of the genes are described by their color. So for instance, those in purple are involved in xylem cell specification. Dark blue are those involved in cellulose, red lignin, and so on and so forth. So there's 209 genes and 497 interactions within the network. Every single gene within this network is expressed within the xylem cell. Also, although you can't see it in these edges in the network, we've assigned a weight to the edges, which are based upon how many reporters were activated in the screen, as well as if there was evidence of these interactions occurring in the literature. And we've identified a large number of the interactions that have also occurred within the literature, which helps validate our approach. Okay, so I'm going to focus on a subset of these interactions, which help to answer some of those questions that I raised before. Okay, so the first question was, what genes are responsible for setting up the timing of this process in the root? So first, an additional note about Arabidopsis roots. So Arabidopsis roots go through, or every single cell within the Arabidopsis root goes through 3 additional developmental programs. So first, after they're produced from the stem cell niche at the tip, they go through the meristematic zone, which is characterized by rapid proliferation. They then move into the elongation zone, where they elongate parallel to the root's longitudinal axis. And they then move into the maturation zone, where many cells terminally differentiate. So secondary cell wall biosynthesis, or the genes associated with this process, are often upregulated at the boundary between the meristematic and the elongation zone. So in the meristematic zone, like I said, it's characterized by rapid cell proliferation, or mitosis, where you're going through G1 to S to G2 to M. And cyclin B1 is often a marker of cells that are undergoing mitosis. And when these cells enter into the elongation zone, many of them undergo endoreduplication, where they basically go from the G1 to the S to G1 to S. And this is all determined by the activity of a transcription factor called E2FC. E2FC inhibits the transition from G1 to, from S to G2. So the cells basically go and endoreduplicate. So why are we interested in the cell cycle? Well, in the yeast network, we found that E2FC binds to the promoters of VND6 and VND7. So we were really excited about this because if you look at where E2FC is active, so E2FC is active right at the boundary between this meristematic and elongation zone, and VND7 is turned on at the boundary between the meristematic and the elongation zone, as is VND6. I'm just going to be showing VND7 pictures in this slide. So we predicted, based on this model and this temporal information, that E2FC must be activating VND6 and VND7 expression. But as with all models, sometimes your prediction can turn out wrong. And so yesterday, in fact, we were looking through this data and we realized that, so in an E2FC RNAi mutant, you can see that VND6 and VND7 expression is upregulated, which indicates that E2FC is acting as a repressor of VND6 and VND7. Okay, so how does this work? How do we put this all together? And most importantly, what is keeping VND7 off in the meristematic zone or turning VND7 on as it goes into this boundary into the elongation zone? So in collaboration with François Roudier at École Normale Supérieure in France, we had previously set out in a project where we sorted vascular cells that are specifically in the meristematic zone, and we did chromatin immunoprecipitation coupled with microarray analysis to look for genes which were targeted by H3K27 trimethylation. And we did this because genes that are targeted by H3K27 trimethylation often show a decrease in their gene expression, or they're silenced. And so in fact, when we looked at this data set, both VND6 and VND7, again, I've just shown VND7 here, show a statistically significant increase of H3K27 trimethylation marks in both the upstream regulatory sequence and along the gene body. So this is consistent with H3K27 trimethylation repressing the expression of VND6 and VND7 in the meristematic zone. So great. So what about E2FC then? So we predict, or we hypothesize now, that E2FC perhaps is responsible for repressing expression of VND6 and VND7 specifically in metaxylem cells because there's a delay in the differentiation between metaxylem cells and protoxylem cells. And so this is a hypothesis now that we're testing in Planta. There are also several other transcription factors which were found to bind to the promoter of VND6 and VND7. So we've now gone back to that data, and we're testing the hypothesis of whether or not any of these three transcription factors can activate the expression of VND6 and VND7 at the right time. Based on the temporal patterns, a MIB is our best candidate for this. Okay, so now moving on to the connection between these transcription factors and enzymes associated with secondary cell wall biosynthesis. So we had found that VND7 binds to the promoter of SES7, which is associated with cellulose synthesis. And it had been previously published that in a VND7 inducible line, that SES7 was in fact upregulated. So this is consistent with our hypothesis that VND7 acts as an activator of cellulose synthesis. Okay, so now moving on and elaborating upon this module. So what's the order between these NAC domain transcription factors and these HD-ZIP3 transcription factors? So we found that VND7 binds to the revoluta promoter. And so based upon the temporal expression data, we found that revoluta is expressed most highly in the roots meristematic zone. And again, VND7 is expressed later on at the boundary between the meristematic and the elongation zone. So this is consistent with revoluta acting as a repressor, with VND7 acting as a repressor of revoluta expression. And so we, in fact, have CHIP data suggesting, in fact, that VND7 in Planta does bind to the revoluta promoter. And in a VND7 induction line, we also showed that VND7, in fact, does act as a really potent repressor of revoluta expression. Okay, so now let's move from revoluta expression to enzymes associated with lignin biosynthesis. So we found that revoluta binds to the promoter of 2 enzymes or 2 genes associated with lignin biosynthesis. And in a revoluta mutant background, you can see that there's an upregulation of these 2 genes, again suggesting that revoluta acts as a repressor of lignin biosynthesis. So putting this together, how does it all work? So we have an H3K27, H3K27 trimethylation of VND6 and VND7, a cell cycle regulator that somehow represses VND6 and VND7. But most importantly, VND6 and VND7 repress the expression of revoluta, which thereby relieves the repression of these enzymes associated with lignin biosynthesis, making sure that lignin biosynthesis will turn on at exactly the right place and the right time. And then VND6 and VND7 in parallel can then also regulate or activate the expression of enzymes associated with cellulose biosynthesis. Okay, so moving on to other ways in which we can analyze this network. So as you can imagine, that network is a really, really large, complex, interconnected network. When people look at that network, they sometimes get scared, especially members of Mallory's committee. And so we, and maybe Mallory herself too, and so we needed to find ways to be able to reduce the complexity of this network. And we did that by collaborating with a physicist named Sebastian Onner. So what he did was he developed a compressibility measure, which basically looked at all the information specified by each of the edges and tried to see if he could compress any of that information into single units. So using this measure, he was able to identify these different modules or network neighborhoods that we predict will regulate different aspects of secondary cell wall biosynthesis. So for instance, there are 13 TFs that bind to the promoters of 3 enzymes associated with lignin biosynthesis, 3 TFs that bind to the promoters of genes associated with cellulose biosynthesis, and so on and so forth. So the prediction is that you should be able to start to make pairwise combinations between these transcription factors and then start to see a corresponding reduction of lignin, let's say, or cellulose biosynthesis. And this is likely the reason why, in many cases with these transcription factors, if you have a single loss of function mutation, you don't see an influence on the metabolic phenotype. And so these different modules or neighborhoods are predicted control points for Xylem development and secondary cell wall synthesis. Okay, so as an example here, so a loss of function mutation between these 2 TFs, which on their own, loss of function mutations between both of these 2 TFs shows a corresponding decrease in the amount of lignin produced in the leaf. So now Mallory could theoretically go and start making all the pairwise combinations between these genes and see which ones results in a reduction of the amount of lignin. But that's still 13 TFs, and you can imagine all the pairwise combinations, and that's still a pretty tall order. So what we decided to do was to also incorporate additional computational modeling approaches. So first, we looked at 2 different computational approaches. The first, which looks at the putative regulation of a TF based on its target in time. The idea here is that a transcription factor can putatively regulate the expression of its target in time. And if so, then this TF and its target likely show similar shapes of their expression profile, but just shifted in developmental time. We also employed a Boolean modeling approach. And then we also looked at data looking at gene expression in the root in specific cell types in response to stress. And so the idea here is that we know that if a plant has a reduction in the amount of cellulose or the amount of lignin, it is in fact very sensitive to external stresses. So our prediction was that perhaps in response to these different stresses, this underlying gene regulatory network might be changed, so the genes within this network might show differential expression. And so luckily, there are data sets of the same vascular meristematic tissue in response to a wide variety of different stresses. So in response to iron deprivation, salt stress, pH, sulfur, and nitrogen. So when we looked at the genes within our network and looked to see if any of them changed specifically in vascular tissue in response to these stresses, 3 of them did. So iron deprivation and salt stress largely activated a lot of these genes in the network, while sulfur repressed many of these genes within the network. So our prediction here is that specifically for iron deprivation and salt stress, for instance, that if you expose a wild-type root to this stress, you may activate the secondary cell wall biosynthesis program. And Ali in my lab tested just that by looking preliminarily in response to iron deprivation. This is a wild-type control route, and you can see here that this is the point where xylem cell differentiation starts to occur. And we stained these roots in the control plants and in wild-type in response to this iron deprivation stress with a stain that marks lignin, so fluoroglucinol. And so you can see here that xylem cell development begins precociously, and you also can get ectopic lignification. So this iron deprivation is sufficient to activate this xylem cell development and differentiation program. Okay, so using all of this data, so there's additional time course data for the response to iron and salt, we were basically able to take that set of 13 genes, which regulate the promoters of enzymes associated with lignin biosynthesis, reduce that set of 13 now down to 6. And so this is a much more reasonable set of higher order mutant combinations to make. And so just to summarize what I've told you so far, so these enhanced yeast-1 hybrid assays provide a platform to rapidly map Arabidopsis gene regulatory networks, not just Arabidopsis gene regulatory networks, but also gene regulatory networks of any organism. These approaches have been used in human, worms, and in flies, and they're also really amenable to any plant system. All you need to know is which transcription factors are expressed in which tissues, and you need to identify a set of genes and be able to at least clone a KB upstream of their promoter. The Xylem cell specification gene regulatory network is highly combinatorial, which reflects the well-known observed genetic redundancy within the Xylem cell specification and secondary cell wall biosynthesis network in plants. We've identified direct transcriptional connections between developmental specification genes and enzymes associated with metabolism. We've identified suites of genes which are potential control points in xylem cell specification and secondary cell wall synthesis. And this xylem cell specification and differentiation network is highly responsive to stress. And so there are several people to thank. So Mallory is the graduate student who started off on the xylem cell differentiation network. Ali is the person who developed the enhanced yeast-1 hybrid assay and who looked at the stress responses. Ted is a graduate student who is performing a lot of the bioinformatic analyses in our lab, and the chip data was produced by Francois Roudier and also by Miguel De Lucas, a postdoc in our lab. This has been a really great collaboration with Doreen Ware's lab and her group at Cold Spring Harbor, the lab of Marion Walhout at UMass Med, Sebastian Aner at Cambridge, Steve Kay, who assisted us with the transcription factor collection, Francois Roudier, and other collaborations that I didn't speak about today with Sam Hazen and Terry Long. I'd like to thank funding from NSF and our other funding sources. So thank you, and I'll take any questions.
Questions for Siobhan? Jim?
I was struck that the xylem cell differentiation regulation appeared to be primarily negative, a lot of repressive effects and not much in the way of positive regulation, which is quite distinct from what I'm used to looking at in mammalian development.
Right.
Do you think is there biology behind that, or is?
So I think, and I mean this is just completely speculative, I think that it's because you want to make sure that these compounds or that the expression of these enzymes are turned on, again, at the right time or the right place. So it's better to have many different fail-safe mechanisms which turn them off until the right point at which you can relieve these multiple repressive lines. And I think it's also actually been found in development of other organisms as well, that you have these multiple repressive steps.
Okay, I was curious about heterogeneity for like expression profiling. Are the tissues pretty clean, or do you get mixed up signal because of heterogeneity?
Right, so in terms of like the original data set where we profiled expression in 19 different cell types, in some cases, the cell types that we profiled were combinations of cell types, but in some cases, they were just an individual cell type. When it was just an individual cell type that was being marked by GFP, we were able to go back, look at the cells that came out of this order, and see that they were all that same cell type. So we had like, I think our percentage of purity was like usually 98 to 100%. But you really need to have a talented FACS technician.
Okay, well, thank you very much. We're going to keep going to stay on time.